Dear friends, I am Dr. Hemlata, Biology faculty. We are going to start with the second PU syllabus because it is required for you to start with the first unit itself, that is about the reproduction. So today's chapter is reproduction, and you know there are 4 units, subunits under reproduction. One is reproduction in human beings, then reproductive health. Third one is the plant reproduction. Fourth one is reproduction in organisms. But actually in your NCERT syllabus they are given first chapter as reproduction in organisms, where they deal with the details of general reproduction. But as we are talking about the importance of the chapter, we have taken up the first unit as reproduction in humans. Okay, let's begin with reproduction chapter and detailed explanation of human reproduction I'll give you. Let's start with the first one. What is this reproduction? Right? Everybody you can understand, it is the production of offsprings by the parents is called reproduction, right? Parent or parents, two things I'm using. Parent is uniparental reproduction, normally we call it an asexual reproduction. So in lower organisms, we have asexual reproduction, and sometimes a vegetative propagation, single parent can be involved. But most of the higher organisms, reproduction is sexual. That means the male and female organisms are different. There is sexual dimorphism observed, right? So we are going to talk about reproduction in humans as it is. It's a common understanding that humans undergo sexual reproduction. So then what is sexual reproduction? That is involvement of two parents. One is male parent, one is female parent. They produce the male and female gametes respectively. These gametes fertilize and produce a diploid zygote. Zygote will grow as an embryo. Embryo will develop into egg. This is how the sexual reproduction pattern follows. So, but when you talk about the human reproduction, before entering into the reproduction process, let us learn about the structure of reproductive system. So, humans, as you all know, they are sexually different. They are showing sexual dimorphism. Male and female, both of them are different appearance-wise, genetically, physically, physiologically. Therefore, we have to learn about male reproductive system separately, female reproductive system separately. Now, when you talk about the reproductive systems structure, we have two important things. One is primary reproductive structure, secondary reproductive structures. Primary reproductive structures are those which are directly involved in gamete formation. They are normally gonads. They produce the gametes. Secondary reproductive organs are those which are helping for the formation or the movement of gametes and then the process of reproduction. So imagine we are talking about the male reproductive system. The major primary reproductive structure in males is only the testes. They produce the sperms. This is the male primary reproductive structure. But secondary reproductive structures, as I start drawing it, you will understand what are the other things which are involved in the reproductive system in males. Then in females, the major reproductive system is ovary that produce the egg. Right? Egg is the female gamete. Sperms are the male gametes. Again, in female reproductive system, there are a lot of secondary sexual characters or sexual organs are found. Okay, so before entering, I am giving so much of introduction just because you should have a common idea how the reproduction happens in humans. In human females, if you think of, or males only you talk about, primary, secondary reproductive structures I will give you while explaining. But apart from that, we have accessory glands or accessory structures which are helping for it. Accessory glands are there. And apart from that, reproductive secondary sexual characters. Sexual reproductive characters are those characters that appear when the male or female enter their puberty. Puberty is nothing but the sexual maturity. When the males from the age of 13 to 14 years of age, they enter the reproductive age. We call it as the sexual maturity. So they develop certain secondary sexual characters. What are those? Like males will develop the facial hairs, like moustache, they develop beards. Apart from that, normally you would have seen there will be a thick hard structure in the neck area, that is nothing but laryngeal prominence. That is one important character, and their voice become more deeper. Means they have a little broken or hard voice, and they start breathing from the diaphragm. That's called the diaphragm breathing, right? Therefore, their voice become more deep and hard, right? And also they develop pubic hairs and the body hairs. This is a major character, and they become muscular, and they'll become little taller, more taller during that time, that period, right? But what about the female reproductive, secondary reproductive characters, secondary sexual characters? Females will develop, apart from egg production, they develop menstruation. Other characters are, they have enlargement of breast and enlargement of the pelvis. Then apart from that, body starts secreting more and more fat, and their voice become more thin and shrill, and they will have sternal breathing. Means they will have external breathing, very little, they will not go deep in breathing like how men will do. That is the secondary sexual character in females. Now let us begin with the reproductive system in men and women. Okay, the male reproductive system, first I will draw, then we will talk about the female reproductive system. I want to draw it because you need to draw it in the board, because there will be a 5-mark question, compulsory question, to draw, label the parts of male reproductive system in human or female reproductive system in humans. So you just learn how I am drawing, right? Because you observe how exactly the organs, parts and all I draw, how I will connect them. Therefore, you have to watch drawing, and after I finish drawing, later you pause and practice drawing, right? Because drawing is very important for biology, as you all know. It need not be very artistic diagram, you draw it accurately, correct diagram, right? So let us begin with the human male reproductive system, right? Let me start drawing this, okay? So it is about the male reproductive system, right? So it begins with the two structures on either side. These are the two testes. Two testes I am drawing. They are found hanging outside their body. Then these are present within some membranous sac we call the scrotum. Within this you get the seminiferous tubules, and these seminiferous tubules are involved in the production of sperms, right? They release their sperms into one more glandular structure, we call this as epididymis. This is the epididymis. This epididymis will move upwards, right? Like this, both the sides will be a epididymis, and this is how it is entering. And this is the Vas Deferens. Singular is Vas Deferens, plural is Vas Deferentia, right? Make it as a thin tubular structure, okay? Like this, and both of them they will join what we call it as a urinary bladder. Here is a urinary bladder, above this is the urinary bladder, and here comes urinary bladder, okay? And then this will move down and join the ejaculatory duct, and on this actually you have got seminal vesicles. These are the two seminal vesicles. Then this is how they are releasing. Then comes the duct called as the ejaculatory duct. This is called the ejaculatory duct, and from here actually what we have is ureters. These are the ureters. This is the urinary bladder. This is the ureter. This is the seminal vesicle. Okay. Then we have the gland here. This is the bigger gland, we call it as the prostate gland. Then it will enter the two smaller glands on either side. This is called as the bulbourethral glands or copper glands. Then they will continue in a form of urethra. This is a urethra. I am drawing a urethra. Okay. Then urethra will lead to the last portion, that is penis. What I am drawing is a penis structure. Okay, let me draw this aside a bit. So this is the penis structure, right? Here is the body cavity. Okay, like this. So this is the male reproductive system. So let us label all these parts. So this part, what I have drawn here is the testis. The whatever sac I have drawn here, this sac is called as a scrotum. And this upper part where they are all releasing, these are the seminiferous tubules inside the testis. This we call it as epididymis. Okay, and this is called Vas Deferens. Plural is Vas Deferentia. Singular is Vas Deferens. Then they are all joining here. This is seminiferous tubule. This is called seminal vesicle. They are opening here into the common duct. We call this as the ejaculatory duct, where urinary bladder also opens and our seminal vesicles or the vas deferens also opens here. And this part is called as the gland, right? I told you this is called the bulbourethral gland and this is the another gland. Okay, so this is the bulbourethral gland. Okay, and this is the urethra. This is penis. And the tip of the penis is slightly swollen part, right? Like this. It is the last part of it. This is the urethra tube, common opening between for the urine and sperms, right? This is the tip of the penis. Okay, and here you will find some skin foldings will be there here. These are the foldings of the skin which are normally present in the penis. So penis is the duct or the gland, accessory gland that helps to release the sperms in female genital tract. So you have to remember the structure complete, and the last portion we call this is the glans penis. Okay. So this is the structure what I am drawing. You have to remember this and understand how to remember the parts, right? What all I told you is first is testis, two testes on either side. They are hanging outside. They will be present within the scrotum. Scrotum is a sac in which they are found, and why they are found outside the body, they are hanging outside the body is in that area the body temperature is lower about 2 degrees centigrades because sperm viability is happening only at that lower temperature. Therefore, you have to remember that the testes are not inside the body cavity, they are outside the body cavity within the scrotum. Then within this there are a number of tubules found. These are called seminiferous tubules that produce sperms, and then these seminiferous tubules open into a small glandular duct we call it the epididymis. From epididymis, sperms are carried through vas deferens and open into the urinary bladder area into the ejaculatory duct. This urinary bladder, where the urine is stored. Urine is stored. That's a collecting structure because ureters bring the urine and stored in the bladder. And in men, both urinary tract and the genital tract are together passing. Therefore, in ejaculatory, both of them they open, and there are two glands. This is called the bulbourethral gland. There is another gland found here. Then they will all release the sperms into urethra, and urethra leads to the penis, and penis is the what you call mating organ, or it is the one which carries the sperms to the female genital tract. Okay, just notice the drawing. For some time you just pause and observe the drawing. Okay, students, we have learnt the first diagram showing the front view of male reproductive systems, right? And I explained you the details of the accessory ducts and glands also. But sometimes they will ask you a question to show the male reproductive system in a side view, right? You need to understand how to draw it. First, let me draw and you just go through it. That's about the male reproductive system in side view. Okay, system. I am giving you the side view, how it is appearing if you observe the side view. Okay, first let me draw the testis. Testis containing seminiferous tubules. You need not too much details and all you need not to give, because only when you are giving the details of the accessory glands you have to give this. This is the testis, and the surrounding is the sac. We call it as the scrotum, right? Then comes, this is the epididymis, where the sperms are collected. From epididymis, the sperms are transported through the vas deferens. This duct you have to draw very neatly, because this diagram needs some patience also. And this is the one. It takes around the structure of urinary bladder. Here is the bladder. So it will take one round around the urinary bladder and it will come and join another duct. This is called the ejaculatory duct, where you will find the seminal vesicle. Then below this you find, this is the ejaculatory duct. This is the prostate gland, and here are the bulbourethral glands, and this will transport the sperms along with the urine into the urethra. This duct you have to draw very neatly, because most of the time the diagram carries the beauty of the diagram is important for you. You should draw it properly. See, I am showing you the same reason how to draw it. Otherwise, mostly if I put one ready diagram, you will not be in a position to copy that. So here is the penis. This is the glans penis, and here is the major part of the penis. This is the penis. This is glans penis, the swollen portion at the tip. And this is the major part of the penis. Okay? So this is the front part and this is the back part of this male reproductive system. Okay? So what are other parts? Let me label. This is the testis. This is the scrotum. And the part which is here, which is like a gland, we call it as epididymis. And this is Vas Deferens. Okay. Then this Vas Deferens turn around the urinary bladder. This is a bladder. This is the seminal vesicle. Okay. And this is the ejaculatory duct, where all of them are opening. This is the ejaculatory duct. And the gland which is present here is a prostate gland. Prostate gland. And this is the bulbourethral gland. Okay. And then this is the urethra. And this is penis. And the tip of it, we call it as the glans penis. And here you can just show in, just for the significance, show that this is the foreskin. Okay, this is the structure in the side view. Okay, sometimes they may ask you to draw and explain the side view of male reproductive system. So you are supposed to draw this. And I think I have already told you about the various accessory glands, like seminal vesicle producing the semen, which is a viscous like fluid, and prostate gland is producing another fluid which is nutritive in nature, and bulbourethral glands produce the pre-ejaculatory fluid which will be passing and cleaning the urethra, and it will save the sperms from the acidity of urine. If the urine, because it has a common duct, right? The duct through which urine also passes, the sperms also pass. Therefore, the bulbourethral glands will produce a pre-ejaculatory viscous fluid that will clean the transparent fluid. It is clean up the urethra so that the sperm, and also sometimes dead sperms will be deposited. That and all will be cleared off as the sperms pass through this. Then this is a regular structure. You should practice the drawing of this also. This is the side view. Okay. Now we will move on to the next part, that is about the tiers of the testis. This is only external view. In transverse section we are going to learn at least the growing cells and how exactly sperms are produced from each of the seminiferous tubules from the germinal epithelial cells. Because this structure we will be getting it in the practical examination also. So let us start with the next diagram, that is about the transverse section of testis. Very important question it is. This is a practical oriented question also. So remember, for this you can use little light colored things also. Then it will become more not only impressive, but it should be sharp drawing. Don't shade too much. These kinds of things you can shade, but whenever you draw the histological diagrams, the cell details you are drawing, don't shade the diagrams too much, because every cell has importance in this. So they understand how to draw it. So in testis section, I'll just give you one outline. This is the focus. I'm just showing you in one focus how many you can see. This is one focus of this structure. Okay. Then in this, let me give you the details of the parts. Okay. So let me give you once again the outline I'm giving you here. Okay. In this one or two seminiferous tubules, I'll enlarge. This is one of them. This is another one, half I can draw here, okay? And here we have the outer covering of the seminiferous tubule, like this, and they are having germinal epithelial cells, and you must be knowing one thing, epithelial cells, if they are germinal epithelial cells or any epithelial cells, they are located at the basement membrane, right? So this may be a basement membrane. Then lining this you find number of such cells. These are the germinal epithelial linings. This is germinal epithelial cell. Okay. This is a lining having the nucleus in them. You need to draw the nucleus also here. So it takes some patient way to draw it. Okay. Patiently you should draw this diagram, because this carries marks for you even in the practicals and your records and all. Records you can draw directly from this. Okay. This is the germinal epithelial layer lining. Then first thing is, they start dividing. These cells begin to divide, undergo mitotic divisions. And initial division is mitotic cell division. These will form number of cells. They are haploids, they are diploid cells. They are all undergoing cell division like this. Okay? Like this. They are dividing. Okay? This is the mitotic cell division. Now, these cells are called as the first formed ones. They are the spermatogonial mother cells or spermatogonia, right? These are the spermatogonia or spermatogonial mother cells. Now they are going to enlarge in size. The enlargement, I am just drawing like this, they are enlarged cells. Like this, they are enlarging. These are called primary spermatogonia, and some of the cells will enlarge too much, like this. These are the enlarged cells I am drawing. Some of them are enlarging very big, somewhat like this. These are called as the secondary spermatocytes. These are the secondary spermatocytes. Some of them are enlarging. They are called as the Sertoli cells. Right? Then they will undergo meiosis. These cells undergo meiotic cell division and they form number of haploid cells. These are the haploid cells. After growth, they form haploid cells. These are called as the secondary spermatocytes. Okay. Now they metamorphose to form haploid sperms. These sperms are tadpole shaped like this. then these should be normally attached to the Sertoli cell. The Sertoli cells I draw, these are the sperms, these are the Sertoli cells. Okay, let me label all of them now. Whatever I am drawing here are the sperms, right? Okay, let me label all of them. One by one you understand what I am drawing. The first red one I have drawn is a germinal epithelial lining, germinal epithelium. Then the next layer which undergo simple mitotic divisions, multiple divisions. This layer we call, this group of cells, they are called as the spermatogonia or spermatogonial mother cells or spermatogonial mother cells. These are all diploid in nature because they are just undergoing mitotic divisions. Then the cells undergo enlargement. You can see here the enlarged cells. These form the primary spermatocytes. They are called as primary spermatocytes. These become haploid. These are, I am sorry, they are again diploid. They are enlarged, they grow in size. Very little division, little growth happens in spermatogenesis. They are called as a primary spermatocyte. They are still diploid in nature. Okay, because no meiosis happened. Now the grown-up cells undergo meiosis. Meiosis 1 is a reduction division, meiosis 2 is a simple mitotic division. They form all these cells, which are small cells, what I have drawn here. These are called as the secondary spermatocytes. Whatever we have drawn here are the secondary spermatocytes. They are haploid in nature. After first meiosis, whatever is formed is secondary spermatocytes. Then after second meiosis, whatever is formed, they are called as the spermatids. Now the spermatids undergo metamorphosis. What is the meaning of metamorphosis? The round haploid cells which are non-motile will modify structurally into elongated tadpole-shaped structures called sperms. That we call it as spermateliosis or metamorphosis. Then the next things formed are, all these are the sperms. These sperms are the male gametes. These are the sperms. And they remain attached to the highly enlarged cells. You can see here, these are all highly enlarged cells. They are called as Sertoli cells. Sertoli cells give the nutrients to the, or nutrition to the growing sperms. They are also called as the nurse cells. Right? One diagram, one like this, if you enlarge, other things also can draw like this. Right? Remember structure of testes section. If they ask only, you have to draw this. Right? In between these spermatogonial, or what do you call the total structures, whatever I drawn, seminiferous tubules. Each one is one seminiferous tubule. This is one seminiferous tubule. So each seminiferous tubule is separated from each other. In between you get some undifferentiated cells like this. Okay, these produce the androgens, the male gamete hormones, male hormones, right? These are the modified cells. They are going to produce the male hormone, that is testosterone, right? Remember the structure. These are the ones that are going to produce the male hormone. They are found in interstitial cell area, interstitial area. Okay, observe the diagram and remember and draw it. You can see that these cells, what I have drawn is, they are the interstitial cells. Or they are called as cells of Leydig or Leydig cells. They are important because they produce the male hormone or androgens are called as testosterone. So this drawing is very important for you for practical and theory. Okay, practice drawing both of them and understand. At least keep some rough sheets. Keep on drawing because almost every exam is like a guaranteed question. One of the main part reproductive system will appear. Right? Whether you know the process or not, at least knowing the drawing and practicing drawing, you can get 5 marks. It's easy. So practice drawing without fail. Take pen, pencil, erasers and shading pencils and all. Practice drawing. They are very important for you in second PUC. Right? Okay. Go through them and practice them. now we'll start with the female reproductive system, students, because male reproductive system, almost everything I have explained you, the structure and the parts and the accessory glands and all that. So now coming to the female reproductive system, females normally contain, I told you, the primary sex organ as ovary, and ovary will produce the eggs, right? Normally egg will be produced, and females will have a reproductive cycle of 28 days, during which the egg will be matured from the Graafian follicle and released. We call it as the menstrual cycle, right? Later we will be learning how menstrual cycle is operated, what happens in that. Then normally females, secondary sexual character in females involve mainly the development of breast, already I told you, and pubic hairs and pubis and will develop, and apart from that they will have a shrill voice. Then onset of the female or puberty will start in females by beginning of the menstrual cycle. We call it as the menarche. Menarche is a word which will be asked as one mark question. What is menarche? Menarche is the onset of the female reproductive cycle or menstrual cycle in females at the age of 10 to 12, very early compared to men. It starts. Then at the age of 50 to 55, this particularly menstruation stops, or the menstrual cycle, reproductive cycle stops at the age of 50 to 55. We call it as a menopause. Understand the word properly. The menarche is the onset, the beginning of menstrual cycle. The stop of the menstrual cycle is called as the menopause, where the females will not further undergo menstruation. They will lose the ability of getting pregnant, or fertility is reduced. That's called the menopause. So let us begin with the female reproductive system in humans, right? So female reproductive system. Okay. So this I told you, it is found in the female abdominal area. So let us begin with the structure. Major structure becomes the uterus, which all of you must be knowing, where the fetus will grow by getting the nourishment from the placenta, and the development happens by the enlargement of the uterus during pregnancy. So let me give you the structure of uterus first, then we will go to the details of that. Okay. So uterus I am drawing here. It's a kind of wedge shaped structure, and you keep drawing this kind of drawing, which is carrying 5 mark again to you. This is, first you put the outlines. Here is the fallopian tube or the oviduct. Oviduct will end with the highly opened finger-like structures called fimbriae. On either side I will draw the fallopian tubes. Okay, like this. Then here you find there will be a membrane in which the ovary is located. Okay. Before entering into the ovary, let me give you the parts of the uterus. So this uterus is lined by certain membranes. The outermost membrane, we call it the perimetrium, right? This is the perimetrium, which will continue with this. You keep drawing like this and practice this perimetrium. Then comes highly muscular structure, we call it as the myometrium. This is a highly muscular structure, we call it the myometrium. This is the myometrium. Then highly vascularized structure, we call it as the endometrium. This is the endometrium. Okay, and here is the cervix. Last portion of it is cervix. Okay. Second layer I am drawing, this is cervix. So whatever I am drawing here, it is the endometrium, highly vascularized, lined by the columnar epithelial cells with cilia. This is the endometrium. The lining is called the endometrium. So you are going to continue like this. Structure should be perfect. Okay. Then the lower portion of this will be continued in the form of vagina. This is a vaginal area. Okay, vaginal area. Okay, this is the internal part I am just drawing. Then outside the female reproductive system, the outside uterus, you find the ovary. This is the ovary. One ovary, a pair of ovaries you will get on either side. These ovaries are having number of outgrowths. We call them as ovarian follicles. When the female child is born, by birth around 200 to 250 ovarian follicles are found in each of the ovary. Okay? And then, what are the other parts? Let me give you details and let's label it. Right? The whole thing what I have drawn is the uterus. We call this as womb. Womb where the fetus will grow. Then, the layers I am drawing, this is the perimetrium. This is the myometrium. This is the endometrium. And this is the area where the fetus will grow, right? Then, this part, whichever coming out, this we call it as or here also I write. This is the ovary with the small outgrowths. They are called as ovarian follicles. Okay. Then this whole thing is fallopian tube. Here one fallopian, here is one fallopian tube. Actually the fallopian tubes are biologically called as the oviducts. This whole fallopian tube can be divisible into three parts. The one which is attached to this uterus, we call it as the isthmus, and which is curved one we call it as ampulla, and the last portion is called as infundibulum. So the whole thing is a fallopian tube. So simply you want to write this is called as the fallopian tube. But we have divisible, it is divisible into isthmus, ampulla and infundibulum. And you can see in infundibulum there are a number of finger-like projections that are motile in nature. They are called as fimbriae. Very important is fimbriae. So whenever the ovarian follicles mature, they become Graafian follicles, they produce an egg, and egg will be released in the body cavity. It will be swept by the fimbriae into the fallopian tube. Right? That's very important. Therefore, fimbriae will be having a number of ciliated epithelium. They will be motile, they will sweep the egg into it and it will be passing through the fallopian tube. If the egg comes on the way, it will be traveling in the fallopian tube for the 2 days, almost 48 hours. And if it comes in between, almost like at the isthmus ampulla region, if the egg is here, if the sperms enter through the vagina during the coitus, then if they come in contact with the egg over here in this area only, there will be fertilization. Otherwise there is no fertilization. Right? This actually happens on the 14th to 15th day of the menstrual cycle. Means at the mid of the menstrual cycle, egg will be released by the Graafian follicle. It will be swept into the fallopian tube, and almost in the middle of this, if the egg meet the sperm, means sperms which are released to the vagina will pass all over here, and if they come in contact with the egg in fallopian tube, there will be fertilization. Otherwise there is no fertilization. Right? So fertilization happens. That is a very important question which have been asked many times. Then where exactly fertilization takes place? That happens in the fallopian tube, almost isthmus ampulla region it will happen. Right? And this is fertilization. After fertilization, the egg may come, or fertilized zygote will come and attach somewhere in the endometrium tissue. This is endometrium, and it begins to grow, and there will be a new tissue developed for the nutrition of the growing embryo. It is called as a placenta. Placenta will give the nourishment to the fetus through the blood vessels. Right? So this is the overall female reproductive structure, and this portion, whatever I have drawn here, you can see here it's slightly smaller, narrower structure. We call it as the cervix, and this portion is called as the cervical canal, or we call this as the birth canal. Baby has to pass, the head will pass through this birth canal and it will be pulled out, or it will be moving out during the childbirth, child delivery. It has to pass through the cervix, right? Then this is the vagina. This is the vaginal area. So I hope you understood how to draw this drawing. Again, it's carrying 5 marks. It's simple to draw actually. So what are the parts you have learned? First is the major portion is uterus or the womb, where the fetus will grow, and then comes the extension on either side of the uterus by fallopian tubes. Then fallopian tubes end with the fimbriae. Then the uterus will be somewhat triangle shaped and lower portion will be very much narrower, and we call this as the cervix. Cervix has a cervical canal through which the fetus has to pass, or childbirth have to take place through this, and there is a vagina through which sperms are released into the female genital tract. And in the uterus, details of the membranes you have to draw. The walls of the uterus, outermost perimetrium, thinner one, myometrium is thicker one. Actually during childbirth, myometrium get rhythmic contractions by the help of hormone oxytocin produced by the pituitary gland, and that will help in ejection of the baby. It starts contracting. The myometrium begins to contract vigorously, and then the baby will be pushed out from the uterus. So this is a very important thing. When you draw it, you should know what's happening also. It's not that just drawing, I don't know process. When I explain, that time I will understand. No, whenever you learn the structure, little bit at least you should understand how is the process, right? This is the process where fetus will be growing here during the childbirth. A complete uterus undergo contraction like this, and the fetal head will move down and it will come out through the birth canal, through the vagina. It will be released during the normal childbirth, normal delivery. Then this is the endometrium, which is having columnar epithelial cells with normal cilia and highly vascularized. A lot of the blood vessels are there. This is highly vascularized. Normally, if the fertilization fails, if the egg is not getting fertilized, if it passes as it is, that time it will be situated here and it becomes thicker, and after the ovulation, almost 15 days later, the whole endometrium tissue, which is newly formed along the dead egg, along the epithelial tissue, it will be slotted off and be released through this, that is called the menstruation, menstrual bleeding. What is menstrual bleeding? If the fertilization fails, the unfertilized egg along with newly formed epithelial tissue that is lining or new endometrium tissue formed, along with the blood and blood vessels, all are slotted, broken down, and everything will be released through this and be passing through the cervical canal, through the cervix and vagina. That's called menstrual bleeding. 5 days the females bleed during the menstrual bleeding. Afterwards, what happens? The bleeding stops. One more side, another ovarian follicle becomes a Graafian follicle, start developing with presence of the hormone, the estrogen hormone. Estrogen is the hormone secreted by normally the corpus callosum, later corpus luteum, I'll tell you later. But right now, estrogen helps in ovulation. As the egg is formed here, after 15 days, one more egg is released to the ovary or the ovarian follicle, Graafian follicle, will pass through another fallopian tube like this. Again it may get fertilized. If it get fertilized, it will come and settle here, implantation happens and the fetus will start growing. Right? This is like a regular cycle. If again fertilization fails, 15 days, again this is slotted off and another ovarian follicle begin to mature here. It's like a cycle in females from the day 1 of menstruation cycle, you are the menarche, till the menopause. This is continuous. Only when they will not undergo this cycle is only when they become pregnant. Females, when they are pregnant, menstrual cycle stops. That's an indication. If there is implantation, the pregnancy is there, then there is no next menstrual cycle beginning. That indicates she is already pregnant. And even after childbirth, if the mother keeps feeding the baby through her breast milk, breast is also one of the accessory structure for the female reproductive system. If mother feeds the baby with her breast milk till she feeds the baby, there is no ovulation happening. This is like a natural family planning. Therefore, females will have lactation amenorrhea, we call it. Lactation amenorrhea means it is when she is lactating, mother is lactating, till that time she cannot have the ovulation. She cannot become pregnant. It's like a natural family planning. Anyway, these are the other things you have to understand along with the structure only. So the structure you understand how to draw it, and this drawing will carry 5 marks. Along with so many other things I explained you, the processes that also will be carrying 1 or 2 mark questions that will come as a separate question. Okay, so draw, practice this again, a 5 mark question. Students, once we have finished with the structure of reproductive systems, we are now going to begin with the process of reproduction. So you know that reproduction involves pre-reproductive phases or steps, then reproductive steps, then post-reproductive steps. So today I am going to give you the first part, that is about the pre-reproductive phase. That is, first thing is formation of gametes. Once gametes are formed, then they will have to rate each other. That's the second phase. Then third is fertilization. Then last is how the embryo develops after fertilization. So let's begin with the pre-reproductive phase, that is nothing but the gametogenesis. So how to define gametogenesis? It is a process of formation of haploid gametes from the diploid germinal epithelial cells, or from the diploid, the sperm mother cells or the ovarian mother cells or oogonial mother cells. We call both of them the mother cells as the meiocytes, because they will become meiosis, they undergo meiosis and later form the gametes. Therefore, when I am talking about the gametogenesis, we are supposed to learn what are the steps, how exactly gametes are formed, which cells are forming it. Right? So as I told you, it is a formation of haploid gametes. Haploid gametes refers to the reduction of cellular. There is meiosis happening. Right? So in males, the gamete formed is sperm. In females, the gamete formed is egg or the ovum. Sperm formation is called spermatogenesis and egg formation is called oogenesis. Right? So under gametogenesis, we have 2 subheadings. First is spermatogenesis, another one is oogenesis. So I am going to start with the first part, that is spermatogenesis. So we are going to talk about the formation of gametes in men. Right? This is the major one to understand. So it comes under gametogenesis. So let us not bother about other things. Let us directly start with the process of spermatogenesis. So if we start with spermatogenesis, we are going to begin the process from the inner layer or lining of the seminiferous tubules. So you cannot just directly write about the sperms are formed in the, they are released. No, we are going to talk in the stepwise, sequence-wise fashion. We are taking, suppose you observe the testis section I had given you. Testes are like this, and here are the seminiferous tubules, right? These seminiferous tubules, if you take section, in their lining, sperms are formed from the lining cells called germinal epithelial cells. These are diploid because they are the still vegetative cells only, because they have to undergo meiosis yet. Therefore, when I start with the spermatogenesis, you think that I am talking with respect to internal germinal epithelial lining. So, let me begin with the first step. This is the first lining I am just drawing you. This is the lining of the seminiferous tubule, and these are all called as the germinal epithelial cells or germinal epithelium. Okay? This is the structure of a seminiferous tubule. Right? Now, let's begin with first step. These cells of germinal epithelium first undergo repeated mitotic divisions. Means simple mitosis. Means just cell division is happening without undergoing any change in the chromosomal number. Because mitosis is called as a equational cell division where there is no change in the chromosome number happening. Therefore, here when they undergo cell division, there are number of cells formed all are diploid only, because germinal epithelial cells, if you think of, they are all diploid, they are 2n. Now what I am drawing is the multiple cells that are formed on mitotic cell division. These are all diploid in nature, and these are called spermatogonial mother cells. Right? These are called the spermatogonial mother cells that are still diploid in nature. They are diploid because they are just undergoing mitotic division. And this first phase where the division is happening, it is called as phase of multiplication. So remembering these things are easy. Multiplication is happening where the germinal epithelial cells are undergoing multiple mitotic cell divisions. Number of cells are formed. I might have drawn on 5, 6 only, but hundreds of cells are formed. They are called as spermatogonial mother cells, or they are called as meiocytes. They are going to undergo meiosis later. Now, second, this is the first phase in this. Second is one of them I am going to enlarge. The same way all other cells also are enlarging. So in the second phase there will be enlargement of this cell, which is spermatogonial mother cells, or we call this also as spermatogonia. Spermatogonia is a diploid cell which is formed by the germinal epithelial cells on multiple mitotic cell divisions. Now it will undergo enlargement. You remember one thing, whenever I am writing about the spermatogenesis, there is no much enlargement in the size of the spermatogonia is seen. There is little bit enlargement, not too much. Like if you observe later, I will explain you, oogenesis, there is enormous increase in the size of the oogonial mother cells, but here it will not have. Just around 10 to 12 folds it is increasing, that's it, and it will accumulate a lot of other cell material, cell components like mitochondria and other things. And this is the second phase, we call it as phase of growth. Right? First phase I told you, it is the phase of multiplication, second is the phase of growth. Now the grown up cell, now we call it as the primary spermatocyte. This also is diploid in nature. Remember, it is diploid structure because there is no meiosis happened yet. Therefore, primary spermatocyte also is a diploid structure. This is a very important question for 1 mark question and all. Whenever you just go through any of the papers, they will say just word. Primary spermatocyte is haploid or diploid. Most of the time what happens is we don't get that picture. I want you to understand the scheme of this complete, right? Because the whole scheme of spermatogenesis is given for 5 marks in the board examination. Even along with that, this can be given as a split question. Like I told you, which is the haploid cell, which is the diploid cell during spermatogenesis. So they will give 3 to 4 cells. That time you have to identify, or you only have to mention it, right? This is still a diploid cell. It is not haploid because just it has grown in size. Now the next phase is third phase. We call it as the phase of maturation. Very important. What is happening in maturation? Maturation refers in this to the meiotic cell division. Means the grown up primary spermatocyte undergo first meiosis, then second meiosis to form haploid spermatids. Right? Now it will undergo first meiosis. Right? This is meiosis 1. It forms 2 haploid cells. Right? 2 haploid cells. These are haploid in nature. They are called as secondary spermatocytes. These are the secondary spermatocytes, and the number is n. So here they are undergoing meiosis. Here this is diploid still, here it is haploid. So diploid in humans is equal to 46 here. Now it will become n is equal to 23. N is equal to 23. Remember, the chromosome number in human diploid cell is always 46. Including autosomes, which are 44, allosomes there are 2, right, totally 46. But when undergo meiosis, It will form haploid cells. First meiosis is said to be reductional cell division. Therefore, each cell is haploid in nature. Now, in meiosis 2, second meiosis, again a division takes place. But it is normally, as you already have learnt in first PUC, meiosis 2 is a simple mitotic cell division. So, this is the meiosis 2. In this simple mitosis will happen, the 4 cells are formed. All the 4 cells are haploid in nature because already they are haploid, which are having 23 chromosome numbers, and this is called as the spermatid. Each one is called spermatid. Totally they are all spermatids. All are haploid in nature. They are all haploid. N number refers to haploid chromosome numbers. 2N is diploid number. So till here is one phase. There is a formation of spermatid. Is the first stage in spermatogenesis. So from here till here, I write as the phase number 1. It is the formation of spermatid. Now, the spermatid formed right now, here they are formed, they will undergo metamorphosis. Metamorphosis, structural and physical and physiological change will happen, and these circular cells which are formed now will get elongated and become tadpole shaped structures like this. These are nothing but sperms. So these are the sperms. Okay, totally from one spermatogonial mother cells, 4 haploid sperms are formed. They are all haploid. These are the sperms. So this process, last process where the circular spermatids are going to convert into the elongated tadpole shaped motile structures. This process we call it as the second phase. We call it as spermiogenesis or spermateliosis. We can call it as spermiogenesis or spermateliosis. Remember, students, this can be another question. Define spermiogenesis. It is the process of metamorphosis or conversion of circular haploid spermatids into elongated tadpole shaped motile structures called as sperms. This you can separately remember because this whole process we have 2 sub-stages. One is formation of spermatid, second is spermiogenesis, or we call this also as spermateliosis. Spermateliosis is again the same thing. Okay, this is the total story of spermatogenesis which can be given for 5 months. Write a note on spermatogenesis with the schematic representation. So you can see this is schematic representation. You need to draw. So when I finish this, you take one pen and paper, pause it and take a pen and paper or pencil and paper and make the page into 3 sub areas. First area you have to write the steps. Second one should be bigger where you are writing the scheme. Last part should be the name of the cells or name of each stage cells. So this way this only scheme can be given for 2 or 3 mark question. Explain spermatogenesis is a 5 mark question, but they may ask you write a schematic representation of spermatogenesis. This is only for 2 marks they can give you, right? Which is already, it's not like I am guessing it. Just go through any of the papers previously. It is very simply given like this, right? Directly they will ask either scheme or they will ask you explanation. But only thing you have to remember is every stage what is the chromosome number, diploid, haploid and all. See, this is 2N, I have written here, I have written 2N. Here it becomes after meiosis 1, it becomes haploid. Again meiosis 2, mitotic division, again they are haploid. These are the sperms and these are all from here till here they are all haploid. Therefore chromosome number is very important and their names are important. See, for the first one I have written spermatogonial mother cells or spermatogonial SMC. Second, after growth they are called primary spermatocytes. They are again diploid only, they are growing in size. Then meiosis 1 there will be haploid condition because it is a reduction division. They form secondary spermatocytes. Then after meiosis 2 they form spermatids, which is again simple mitotic division. Then there is a metamorphosis where spermatids become the sperms. So this you take it from me, this will appear in examination 100%. Okay, now you don't bother about anything, you are studying leisurely at home, whatever it is more advantageous to you, so that you can work hard and understand it easily. There is nobody to disturb you. You may have to disturb yourself, so don't do that. Understand this, write it and write the scheme. It will be an important and almost probable question to you in coming examination. Students, let's now start with the next one, that is oogenesis. Oogenesis is the formation of haploid egg or the ova from diploid germinal epithelial cells that are lining the Graafian follicle. See, remember, I told you earlier when I was talking about the female reproductive system, when the female baby is born, then it will have two ovaries on either side. On each ovary there are 200 to 250 ovarian follicles are found. Then each ovarian follicle begin to mature. By the age of 10 to 12 they will mature and start releasing the egg from each Graafian follicle. If it starts from one side, left side, then that next month it will be starting from the right side. Right and left, it will be every alternate month there will be production of egg from alternate sides. Now Graafian follicle structure I will give you later. Right now I will give you how exactly eggs are formed during the process of oogenesis. Now as I am concentrating on the human reproduction, I will give only how it happens in humans. In between I may give you a little bit about the other animals also as a side view. Okay, so let's begin with the oogenesis. The first thing what I am giving is, this is the inner lining of the Graafian follicle, right? These are the cells called as the germinal epithelial cells. Epithelial cells, which are diploid in nature, from Graafian follicle. So very important is you have to write properly where exactly it is producing. So it is the Graafian follicle internal lining, because ovarian follicle when it matures become Graafian follicle. The Graafian follicle lining is called germinal epithelial cells. These germinal epithelial cells are diploid, they will have to produce the egg. Right? The first stage, as you already have learnt in the spermatogenesis, it is a phase of multiplication. So the first stage what I am writing is phase of multiplication. This is repeated mitotic division. I hope you know it. So you will get from each cell number of such diploid cells. These are nothing but the oogonial mother cells or oogonia, right? So let me write first only how are they. They are diploid in nature. These are called as oogonial mother cells or oogonia. They are diploid in nature. This is called oogonia. Now, next stage, each one of them have to undergo the enlargement. In humans only one egg is formed. Therefore, I am starting with only one egg. One of the oogonial mother cells enlarge. But in all the other mammals, other animals like birds and all, this is a very huge stage. There is an enormous increase in the structure of the size of oogonial mother cell, around 200 folds will increase. But here as humans, we don't have to have such a huge egg. It will be little bit enlarging, but still it is better than what we drew it in your spermatogenesis. This is the second phase, we call it as the phase of growth. Okay, this is the phase of growth where there is an increase in the size of the structure is happening here normally. In this phase, little bit let me discuss. In the growth phase, if you think of the birds or in the frogs, amphibians, these animals undergo external fertilization, and that is mainly frog undergo external fertilization. Whenever it is producing egg, there is enlargement. It has to pack up the full nutritions in this structure only, because this is normally called as yolk formation. Yolk is nothing but the yellow colored tissue. You would have seen yolk in your hen egg, right? Yolk is a proteinaceous nutrition which has to be packed within the structure. The structure which is formed is still diploid in nature, and we call this as the primary oocyte. This is called as the primary oocyte. The primary oocyte is again diploid in nature, oocyte it is, and then this diploid structure in other animals there is a huge increase, not in humans. Why? I will tell you some logic behind this. First thing is, whenever egg is undergoing externally fertilized, imagine with frogs, eggs are released into the water by female frog. As it enters the water, sperms are released by the male frog. The male sperms have to search the egg and should fertilize. After fertilization, suppose it happens, embryo will be formed. Embryo must have to be nourished by the egg itself, which is already packed by releasing. Therefore, the presence of this yolk is very important for the survival of such animals like the frogs and in the birds and all, right? Because they will have huge yolk form. Therefore, 200-300 fold will be increase in the growth phase. At this phase, there will be enlargement. But in humans, it's not so, because in humans, egg is produced without any yolk. Reason? Very simple. Humans undergo internal fertilization. Apart from that, once the fertilization takes place, mother develops a nutritive tissue called the placenta. Placenta will be connected to the fetus, and whatever nutrients mother's blood is having, it is transported across the placenta to the fetus, and fetus is nourished. So there is no need of egg to have yolk in human. Therefore, in human egg, there is no yolk. Such egg is called alecithal egg. Alecithal is the egg containing no yolk. Right? Now, this is the phase of growth, where is enlargement of the size is seen with all the nutrients deposited in this. Now, next phase is phase of maturation. You all know this. Already I explained you in this previous one. This is the phase of maturation. Maturation refers to meiosis. Right? First stage is meiosis 1. Which is nothing but the reductional cell division. Till here, there was 2n is equal to 46. But now what happens? They undergo reductional division. But remember one thing. During meiosis 1, two cells that are formed that are haploid, fine. But during karyokinesis, haploid condition is brought having two nuclei which are haploid. But during cytokinesis, means division of the cytoplasm, divisions become unequal. Why? Because one of the cell will take the complete nutrients and cytoplasm. Another one will be having only haploid chromosome number, but very little or no cytoplasm. Why? Because one will survive better, others are going to disintegrate. Therefore, the first cell formed here is very little, small one, and this is called as the first polar body or polocyte. This is haploid in nature. Remember, n is equal to 23. Right? Now, the other side, there will be a large cell formed which contain the haploid nucleus and all the nutrients in it. This is also a haploid, n is equal to 23. But this is a functional cell. We call this as the secondary oocyte. This is the secondary oocyte having haploid condition. So meiosis 1 itself is reductional cell division. But what I told you, you have to, without fail, you have to remember this point. The whole of process, you have to remember this point. What is that? During meiosis 1 of oogenesis, the cytokinesis is unequal, whereas karyokinesis is reductional cell division. Reduction means chromosome number reduces to half. So one of the cell formed is non-functional. It is called the polocyte. It just will carry haploid chromosome numbers. But this one is not so. It is functional cell. It has haploid condition. Along with that, it has nutrients also in it, because it is going to be a functional egg in future. Now, another meiosis has to take place. Meiosis 2 will happen here separately, here and here together. They are undergo meiosis 2. This is second meiosis. Meiosis 2. Okay. This again, the first polo body or polocyte will produce two more polocytes. Remember, these are never going to be functional. This is another haploid, this is another haploid. They are called as the secondary polocyte. These are called as secondary polocytes or polar bodies, polocytes. What about this? This is the secondary oocyte producing two more haploid cells. Again, the division is unequal. One of them will become polocyte, very small, minute, non-functional. One is a functional oocyte, because that's going to become future egg. Therefore, there is another polocyte formed over here, but one functional. We have this. This is the oocyte. This is another polocyte. All these are polocytes. This is haploid, which is nothing but the oocyte. Remember these words. This is oocyte, which at maturity becomes the egg or the ova, right? So what do you have to remember in this? Here there is no other metamorphosis happens like how it happens in the spermatogenesis. Now what you have to remember here is, it is the process, oogenesis is the process of formation of haploid egg from the diploid germinal and epithelial cells lining the Graafian follicle. That's the first thing. Next is process of formation. First, the Graafian follicle lining germinal epithelial cells undergo mitotic division, repeated mitotic division. They form large number of diploid oogonial mother cells or oogonia. They are all diploid only. Now it undergo growth phase. There is enormous increase in the size of the oogonial mother cell, forming a large cell which is called as the primary oocyte. But as I told you, this is not too much of increase in humans because they are undergoing internal fertilization. But all those animals which undergo external fertilization, there will be a huge, enormous increase. There is almost 200 folds increase in the size of the structure. Primary oocyte is seen, during which lot of yolk is deposited. Yolk is nothing but a yolk protein is deposited because they want to provide nutrient to the future embryo. Now in humans, there is little increase. Fine. Now phase of maturation is the third phase in which the primary oocyte undergo meiosis 1 and meiosis 2. Now this is the point to remember. Meiosis 1 is a reductional division. You all know that. But in meiosis 1, the division of the nucleus is the reductional type. Chromosome number becomes half. So you will get n equal to 23, n equal to 23. But during the cytoplasmic division, division is unequal. One of them will become very small, and this is called the primary polar body, the first polar body. Next becomes one of them which is functional, it will be called a secondary oocyte. It is large, containing the haploid nucleus and the cytoplasm. Then meiosis 2 is the second mitotic division almost, in which primary polar body of the first polar body undergo simple mitotic division, form two more polocytes or polar body called secondary polocytes or second polocytes. These are all haploid and they are all functional. But what about this? Secondary oocyte also undergo meiosis 2 and form one more haploid secondary polocyte and then one functional oocyte. Oocyte it is going to become in future the functional egg cell or the ova. This you have to remember the scheme and understand where exactly it is unequal division. Very important points you have to note it down. So after I give some time for you to note it down, just pause it and understand all these things. Then scheme, practice the scheme, both spermatogenesis and oogenesis. Then understand how are they differing from each other. Okay, so I want you to first concentrate on the scheme and then explanation. Then afterwards I will give you distinction between spermatogenesis and oogenesis. Okay, just copy the diagram. Students, now after finishing spermatogenesis and oogenesis, we are now going to distinguish between spermatogenesis and oogenesis, which is a 5-mark question to you. So it's like a combination of both. You cannot just remember one I know spermatogenesis, one I know oogenesis. No, I'm sorry. You have to distinguish between both of them. So how to differentiate between spermatogenesis and oogenesis? First thing, spermatogenesis is the formation of haploid sperms from the diploid germinal epithelial cells or the spermatogonial mother cells in the lining of seminiferous tubules in testes. All these things. What are the words you have to remember? Formation of haploid sperms was first thing. Next, from the diploid spermatogonial mother cells or the germinal epithelial cells lining the seminiferous tubules in the testes. This is first. What about oogenesis? Oogenesis is the formation of haploid egg from the diploid germinal epithelial cells or the oogonial mother cells lining the Graafian follicle of the ovary. Correct? Right. This is the first point. Second point. Spermatogenesis involves the formation of 4 haploid sperms from one spermatogonial mother cell. That's the first thing. Right? Formation of 4 haploid sperms from one diploid spermatogonial mother cell. What about the oogenesis? It is the formation of single egg, haploid egg, from a single diploid oogonial mother cell. Did you get the difference? Therefore, gametes are formed from one spermatogonial mother cell. Here, only one egg is formed from the one oogonial mother cell. This is a very, very important thing for you to remember, because this is the only such things are carrying marks to you. Right. Next is, the 4 sperms formed are normally motile and are haploid. They actually move in the female reproductive tract. What about the egg? Egg is non-motile. It is a large structure. It is non-motile structure. This is sperm and ova. Then one more thing, difference is, during spermatogenesis, there is a metamorphosis of spermatids into sperms. There is total structural change is happening, but here nothing. Oocyte hardly will change into, no much changes happen to form egg almost. There is no metamorphosis. Then one more thing. In spermatogenesis, meiosis 1 and meiosis 2 are showing equal cytokinesis and karyokinesis. What about this? In oogenesis, the cytokinesis is unequal in meiosis 1 and meiosis 2, due to which there are 3 non-functional haploid polocytes are formed with one haploid functional oocyte. Therefore, remember where exactly it differs. Okay? Then this is the important thing you have to remember for the difference between spermatogenesis and oogenesis. Okay? Now let's move on to the next last part of the structural thing, that is about the female mammary gland. This is a very important question for you, which has been asked in many of these examinations. So let me give you the structure of mammary gland in females. I hope you know that females in all the mammals, they feed the young ones with the help of their breast milk. Right? This is an important thing you have to remember, because females mammary gland is an important structure for you, which appears for 5 marks. Okay? So let us draw the female reproductive structure. Now, female mammary gland. So how the structure appears to you. right? So first thing, I am just giving you this part. Whatever I am drawing, this is the backbone. Here are the vertebrae, right? These are the vertebrae. You have to draw this. Then after this, there is pectoris majora. These are muscles. Pectoris majora. From here, because the mammary glands are the modified sweat glands, right? So they appear from the first part of it. This is the structure of the enlarged. Females undergo enormous increase in their mammary gland when they enter their puberty, right? And here it will end up with the small nipple. Okay, this is the nipple I am drawing. And this is the lower portion. Okay. This is the small nipple I drawn. Around the nipple there will be pinkish part present. This is highly pigmented structure or red blackish structure. We call it areola. This is a nipple. What I have drawn is a nipple. Now in this I will draw small lactiferous ducts. Right? There are two lactiferous ducts. So whenever you are drawing, you remember that you will have to draw it properly, right? So here, let me draw two lactiferous ducts. This is a nipple structure I am drawing. Here, after this I am drawing the lactiferous ducts. This is one lactiferous duct, this is another lactiferous duct. Okay? This is in the nipple. This is what mother is going to feed the baby, right? And mother has to feed the baby so that baby will remain healthy, because the mother's milk contains maximum antibodies against most of the diseases. Then this is the lactiferous duct. Further will branch into number of lactiferous, almost ossicles we call them as, where the milk will be deposited, right? Like this milk will be deposited. These are called ossicles, lactiferous ossicles. And these lactiferous ossicles will collect the milk from various alveolar ducts. Okay. Further, they are going to branch like this, entering each of the alveoli. It is not much complicate this structure. Okay, like this. Show around 6 to 10 branches like this. Okay. Then this is about the branchings of the alveolar ducts. Okay. Then they will all open, or they are released into this. There is a release of alveolar ducts. These are the alveoli. These alveoli are found in bunches. In small alveolar sacs they are found. This is one alveolar sac. Then another alveolar sac I am drawing here. Right? They are all in the alveolar ducts. Okay? These are all alveolar sacs. They are producing milk. Actually, they are secreting milk. Okay? These are all alveoli found in one sac. This is one sac, this is another sac, the alveolar sacs are here. So I am drawing only the mother who is lactating. Right? In lactating mother, this alveolar ducts, just observe, they will be ducts, they are the open, they are the modified sweating gland, they will be having something like this and they are open. Right? Suppose I draw this is an alveolar duct. In normal people it will be almost like this closed. Right? In lactating mothers, these will open up, break open and release the milk from this. Therefore, whenever you draw the ducts in the lactating mother, they are all open like this. So these are all ducts joining this. They are all alveolar ducts. They are all opening into this. Okay, and this is about the alveoli. But what is the major thing found in female mammary gland is lots and lots of fat. This is very important for you. Fat is highly deposited. This is all whatever I am drawing is a fat. Fat deposition is more in females because it is a female secondary sexual character also, right? This is the female secondary sexual character where a lot of fat is deposited in women, right? So whatever I am drawing, these are all fatty areas, right? Most of this will be fat, right? Right? Remaining, some of them are I am drawing alveoli. Okay? This structure will carry 5 marks to you, right? What are to label? So let me begin with this. This is the nipple. These are the lactiferous ducts. And these are the lactiferous depositing structures. I told you, ossicles. These are the alveolar ducts or mammalian ducts, mammary ducts, and these are the alveolar sacs. And each one is the alveoli. In each one, this is an alveoli. And this is pectoris majora. And this is the vertebral column or backbone, we call it as vertebral column. And the black portions I told you here, it is called areola. A-R-O-R-E-O-L-A, areola, right? This structure you have to remember, draw, label, it carries 5 marks, just drawing and labeling. But one thing, real students, you have to remember, the female mammary glands are so important. Although they are produced in all the females once they enter puberty, only at the time of pregnancy they start more secreting, producing the milk in the mammary glands, and at the time when the child is born, there will be a hormone release called oxytocin. Oxytocin is called the quick birth hormone. It helps to push the baby out during the childbirth. It, along with there is a lactogenic hormone, these ducts I showed you here, they open up and there is release of milk, and mother starts secreting and releasing the milk through this. Only when a baby sucks the nipple, the milk will enter into it. And you must know very important thing is, dear students, you all have grown up like that. That's a thankful thing about the motherhood, and mother will feel happy to do that, and mother has to feel this because she has to feed the baby up to 1 year, because up to 1 year this baby's intestine has pores in it. Whatever mother's antibodies and all important proteins are given, they are directly absorbed up to 1 year. So mother has to feed the baby up to 1 year, and even during then she will have natural family planning. She will not undergo any ovulation, that is no egg formation, no second pregnancy immediately. Therefore it's called lactation amenorrhea. Mother has to feed the baby, and only such children will have very good development of the brain. Right? Those children who drink milk from the mother's breast, they will grow very nicely, very intelligent. And lactating mother per day produces 1 to 2 liters of milk. Remember children, the mother will produce 1 to 2 liters of milk. So continuously she can feed the baby till from morning till night, night to morning, everything. But one thing is, during this you can see the cells are broken down, mammary cells are broken down, due to which a lot of cytoplasm is lost. Mother will have a lot of tiredness because her cytoplasm is converted to milk and it is fed to the baby. That's the reason why mother has to be given a lot of nutrients when she is lactating. She should have a lot of proteins, a lot of vitamins, a lot of carbs, everything. She needs a complete balanced diet, and over-nutrition is also required for her. So you note down the diagram, practice drawing, and understand this is the must for your life also and for your examination also. Okay? Learn the drawing and draw, practice drawing. Okay? I'll give one minute for this.